Good day, everyone. Today's revision, we'll be talking about properties of amino acids. Is considering the properties of amino acids, we'll consider both physical and chemical properties of amino acids. Now, starting with the physical properties of amino acids, we'll talk about only three properties. The first one is solubility, which we've actually talked about in our previous revision class as the last thing we discussed, where amino acids were classified on the basis of polarity. And the polarity is based on its ability to interact with the ions produced inside the water. So some can interact while some cannot interact. Those ones that cannot interact, we are getting as non-polar amino acids, where we have an example of glycine, leucine, isoleucine, valine, tryptophan, etc., etc. And then we have from the other side the charged amino acid, which could either be the positively charged or the negatively charged, as well as the polar amino acid. Those amino acids that contain within their R group are very, very highly electronegative atoms like oxygen acid 7, like a sulfur acid 16, etc., etc. Now, another property of amino acid is taste. Some amino acids taste very sweet, like glycine, which we give example during the introduction class. Another amino acid like leucine and isoleucine are very bitter. Amino acid like valine and methionine are tasteless. Amino acid like glutamate, we say they have this umami taste. That is why they are used in the synthesis of monosodium glutamate, the normal malt that we all know. The next is light absorption. Some amino acids like the benzene ring containing amino acids like phenylalanine, tyrosine, and tryptophan absorb light at 280 nanometers. This amino acid made it possible for a protein to easily be detected within 280 nanometers in the sample. So if you have a protein in your sample, you can be able to know by that sample absorbing light at 280 nanometers. It simply tells you that there is a protein. And these absorptions are classified into the presence of these benzene ring containing amino acids, which are phenylalanine, tyrosine, and tryptophan. So these are the ones we can talk about. You can also read more and find other properties or physical properties of amino acids. Now, the next we'll talk about is the chemical properties of amino acid. Amino acid can undergo a lot of chemical reaction. Some of them is the formation of esters, where we mentioned formation of monosodium glutamate. We have an example of formation of monosodium glutamate. Decarboxylation, where the carboxyl or the primary carboxyl group can actually be removed. For example, in histidine to form histamine, in tyrosine to form tyramine, etc. We have transamination, where an amino group of one amino acid can be transferred to another substance like alpha ketoglutarate or alpha keto acid generally. Then another property is the acid-base property of an amino acid to discuss. We talk about how an amino acid can behave as an acid, can also behave as a base. And we said the classical the definition is when you have a molecule having the potential to release a protein into the environment, that is regarded as the classical behavior of an acid. An amino acid has that. If you have a compound that have a classical behavior of accepting a protein from a solution, of accepting a protein anywhere, this is the classical behavior of the base site. Now, amino acid also have this altitude. So, both the carboxyl group is the one that has the potential to release the proton, which is acidic in nature, and the amino group, which has the potential to accept a proton to form an ammonia. So this is the behavior of an amino acid. This behavior is called amphoteric. That is what made an amino acid to be regarded as an ampholyte. At isoelectric pH. At isoelectric pH is the pH at which an amino acid will acquire an equal positive and negative charges, so that that amino acid is considered neutral. So that pH is called isoelectric pH. And where we talk about the isoelectric pH can be determined. And how do you determine the isoelectric pH? Is to add the pKa of the two amino acids and then divide by 2. Sorry, the pKa of the two group, the amino group and the carboxyl group. For the amino acid that contain only one amino group and only one carboxyl group. So it is easy to do that, as also like we have explained how that can be done using titration, where you have the solution of the amino acid in your test tube, and then you titrate against the sodium hydroxide, where as you keep adding sodium hydroxide, you are raising the pH. You reach a point whereby the pH will no longer raise, so that the pH is equilibrated. You are reaching an equilibrium between the amino group, an amino group that contain the NH3 and the group that contain NH3 plus and the group that contain NH3 is becoming at what do you call it, at equilibrium. By the time you keep on, you also reach the basic pH. At the basic pH, as you keep on adding the sodium hydroxide, you reach a basic pH. Then you reach a stage where you have the amino group also existing, either as an ammonium or ammonia. Ammonium with the positive charge and as a neutral NH2. So within that, you have this linear. So at the middle of that linear, you have your pKa. So when you want to calculate the pKa of an amino acid, you now add the pKa of the amino group, which is found in the basic region, or the pKa of the carboxyl group, which is found around the acidic region. You add them and divide by 2. That is the isoelectric pH. That is the pH when that amino acid, the positive and the negative acid of that amino acid are equal. Then the amino acid is considered neutral. So if you have any question, please post your question through the class rep, and the question will get to me. The answer will get back to you through the class rep. Thank you very much.